Hello everyone, I'm Christine Rose Smyth, an independent researcher joining you from the Cayman Islands. My goal today is to convince you that this orchid and its pollinators are a fascinating opportunity to answer questions on the role that scents play in pollination by reward and deceit. If I only had this one slide, let me make my elevator pitch to you here. Myrmecophila thomsoniana is an endemic orchid on the oceanic island of Grand Cayman. Note the anolis lizard on the left licking extrafloral nectaries. On the right, observe 3 beetles, one with pollinia attached, a second attempting to copulate with the head of the third. Over 6 years, I've developed a large data set of visitor behaviors, but the technology needed to study floral scent and insect pheromones doesn't exist in the Cayman Islands. I'm grateful, therefore, to SCAPE for this opportunity to reach out to potential collaborators. Please do contact me at the email below if you have chemistry facilities and might be interested in taking on a self-funded student. The white cream-flowered variety var thomsoniana occurs only on Grand Cayman, whilst yellow-flowered var minor occurs only in the sister islands. The rest of the small genus occurs in Central America and Venezuela. Pollination in Grand Cayman is principally by Cetoniinae beetles. The main pollinators from left to right are Genus lanius, native only to Jamaica and Grand Cayman, Protetia fusca, the Asian flower beetle, and Apis mellifera. Additionally, banana quits pierced the side lobes, removing pollinia, as did a female Anolis conspersus lizard once. In later years, rare examples of actual bird and lizard pollination have been observed. 3 other beetle species are occasional pollinators also. And lastly, I'm showing an instance of removal of pollen by the solitary bee, Centris caymanensis. This is significant in that it may indicate that the pollinators of the ancestral orchid in Grand Cayman were bees. This table is adapted from Rod Peakel's Botany 2020 presentation. Ryan Phillips' work with Caledenia pectinata illustrates the important distinction to be made between pollinia removal during sexual behavior and removal during food seeking. In pectinata, the semiochemical attractant occurs on the backs of the long sepals. Male wasps drawn by them get close enough to be induced to switch to food seeking on the lip and result in contact with the column. This concept of uncoupled mimicry is interesting because it poses questions similar to ones that I had asked myself about the behavior of the Myrmecophila beetles. For example, does attraction partitioning occur? Is food reward or deceit aimed more towards the female beetles? Is promise of a sexual reward aimed at the males? Turning to the attractants, yellow arrows point to the source of the only nectar reward, which is produced intermittently. In terms of scent, the lower panels demonstrate that the entire adaxial surface of the labellum takes up neutral red stain, whilst the back of the lip stains much less so. Thus, extrafloral nectaries direct visitors away from the column, but scent can draw them towards it. Focusing on the beetles, the Asian Schaefer is sexually dimorphic, and all Protetia of the orchid appear to be male. Gymnetus displays more subtle dimorphism that is difficult to distinguish in the field. Both species occasionally attempt copulation in or among the flowers. In the top panels, both individuals in each pair is a male. However, most penetrations of flowers are not accompanied by the visible eversion of the adiugus. This may be due not to a lack of action of the semiochemical, but perhaps indicates more complex scent dynamics. Comparing this with uncoupled mimicry then, there seems to be conventional semiochemical attraction in Myrmecophila, but limited uncoupled nectar reward for the beetles. And nectar is the only attraction and reward for the anole. There are many questions that arise from this novel system. One, for example, is, did Thomsoniana evolve a generalized beetle attractant, which explains its ability to attract an advent of alien species? But if so, why is it more effective on Protetia than on other native beetles? My time is up now. So again, if this spikes your interest in a collaboration, please do contact me. And thank you very much, SCAPE and Jeff. Thank you.